Any announcements other than the obvious fact that we have the Apple Pumpkin Bake Off a week from yesterday? 11:30 or something like that?
11:30. Yes.
11:30, great. So think about your recipes. Our speaker today is Dr. Jim Bradeen. Jim is a professor and the head of the Department of Plant Pathology at the University of Minnesota. His background, are you a native of Michigan?
I am from Michigan.
Yeah, and his undergraduate was from Michigan State, and then he went to Wisconsin for a PhD, where he also did a postdoc. He did a lot of specialization in plant breeding and genetics at that time, and genomics, and that led into his coming to University of Minnesota in 2002, and he's been there since. And so he's going to talk to us about this topic today. Welcome.
Great. Well, thank you very much, Mark. And Mark, thanks for organizing this. I want to thank Mosen for the invitation. I think it came from him originally. It's been really a great visit so far. I've appreciated meeting with everyone, and it's a real delight for me to be able to talk a little bit about research, because I don't get to focus on research enough. So I've always worked at the plant end of plant pathology. As Mark mentioned, my background is actually in plant genetics and plant genomics. So I was hired in 2002 at Minnesota to develop a genomics program that focused on plant disease resistance. And ever since then, one of the key hallmarks of the work that my group's done has been utilizing crop wild relatives, wild species that are related to crop plants, and especially gene bank collections as sources of genes for improvement of crops. The research that we've done is really taken multiple different paths. We've worked for a long time on late blight disease resistance. And I think every plant pathology student knows about the historic importance of potato late blight and the Irish potato famine. What you might not know is that this disease today is the #1 biotic constraint of potato worldwide. So potato late blight disease today accounts for about a $5 to $8 billion global loss, either in yield losses or in fungicide applications. So if you think about utilizing gene bank collections or genetics to improve disease resistance in plants, to feed the world sustainably, to reduce the impact of agriculture, potato late blights are a really good model to pursue. And this figure actually represents the Irish potato famine and the historic importance of that disease. So my group has done a series of different research projects focused on potato late blight. We've mapped and cloned late blight resistance genes from wild potato species. We've studied the transcriptional regulation of some of those genes. We've studied the trait expression, disease resistance expression in an agricultural context, and of course we've transferred some of that resistance to cultivated potato for use by breeding programs. But invariably, regardless of the focus of our research, it all started at the same place, and that is the phenotypic discovery of the resistant trait in the first place. So a lot of our research takes place in fields like this. This is a late blight nursery in Minnesota. So the key purpose of this field is to test for disease resistance, late blight resistance in particular, in potato breeding lines, in genetic stocks, and of course in wild species. Now, to give you a visual of what this actually looks like, I'm going to walk you very quickly through a month in the life of a late blight nursery. So this field, #1, receives no fungicides at all. It is an inoculated nursery, meaning that we go out and actually intentionally spray late Phytophthora infestans spores. And in this field, we're actually inoculating every other row. So these are cultivated potato varieties that we know to be disease susceptible. And the idea is that the disease is developed first here, and then spread to the experimental lines that are interleaved between the spreader rows. So we also use overhead irrigation to maintain high humidity conditions, which favors disease development. So this is day 0, the day in which we go out and spray the spore suspension. By day 7, the field still looks pretty healthy from this vantage point. If you actually stuck your head in the spreader rows, you'd see evidence of disease development. By a week later, you're really beginning to see stress, particularly in the spreader rows. By week 3 and by week 4, disease is clearly spread. So if you're a potato farmer in 1840s living in Ireland and your family is almost entirely dependent on potato, you can understand why this disease had such major impact. You can also see from this figure that the vast majority of cultivated potato genotypes are disease susceptible to potato late blight. But there are these islands of green that are throughout this field. More often than not, these are wild potato species. And there are about 200 wild potato species. Many of them are housed in the USDA Gene Bank in Sturgeon Bay, Wisconsin. That's really formed the basis of a lot of our research. This particular species is a wild diploid Mexican potato known as Solanum bulbostanum, and obviously it's a good source of disease resistance. So this approach to trait discovery in gene bank collections or wild species is phenotype driven. Obviously it's successful. We have identified disease resistance in this particular case, but it's also very slow and laborious. Now, plant phenotyping has gotten a lot sexier in the last decade or so. These photos come from my colleagues, Atina Hagik Atalab, Corey Hirsch, and Madeline Smith, all at the University of Minnesota. Essentially, they're using remote sensing to look at plant phenotypes and to do that on a much larger scale. Now, data collection is not the bottleneck here. Data analysis really is, and understanding what from a remote image, what a stressed plant looks like, and especially understanding why a plant is stressed really represents a significant challenge. I think we're making a lot of progress in the area of high throughput plant phenotyping, and this will, to some degree, help us mine those gene bank collections for useful genes at a higher scale. But I maintain that as long as we are still using a phenotype-driven approach, identifying useful traits in gene bank collections is really going to be a bottleneck in terms of crop improvement. Now, to illustrate that point, I'm showing you some research that we conducted a number of years ago on another wild potato species. This is Solanum polyadenium. It's a horrible plant. It's got noxious oils. It smells really bad. It is also incredibly disease resistant to late blight, Vericillium wilt, and nematodes. So it really is a robust species. Now, in this particular set of images, we're testing the ability of this species to grow under increasing herbicide concentrations from 0 herbicide at the top to full strength, label recommended concentrations at the bottom. So clearly this wild species is not a source of herbicide resistance, but in this study I really wasn't looking for herbicide resistance. I was trying to figure out how to grow this wild species that's essentially a weed in our late blight nursery in a way that didn't require hordes of undergrads to grow and weed. So something as mundane as how do you cultivate a wild species really becomes a challenge for trait discovery. Knowing what a resistance phenotype looks like can be a challenge. Solanum polyadenium is morphologically and physiologically very different than potato, and disease symptomology really expresses very differently in some of these species. Of course, in any plant pathology research, the potential for pathogen race specificity is also a particular problem. And all phenotyping, especially phenotyping of gene bank collections, is resource intensive, requiring space, labor, and funding. So all of these really are challenges to a phenotype-driven approach to trait discovery in gene bank collections. So that phenotypic approach is not going to work efficiently for us. What is? Over the last, I'm going to say 8 years, my group has been exploring genotype-driven approaches to inform phenotyping for trait discovery. I'm going to walk you through some of that work that we are doing. Now, to make sense of this research, it's important that we all have an understanding of the molecular basis by which plants recognize and respond to pathogens. Now, plant disease resistance is really a very complicated phenotype, and plants have multiple different strategies for defending against pathogens. But one of the predominant, most important strategies that plants utilize are R gene-mediated disease resistance. And R genes, or disease resistance genes, encode proteins that have certain architectural commonalities. And two domains that we see repeatedly in R proteins are the NB-ARC, or nucleotide binding site, and the LRR, or the leucine-rich repeat. So this NB-ARC and leucine-rich repeat, something that we see over and over again, regardless of what pathogen type this gene's effective against. So it could be a virus, a nematode, a fungus, an oomycete. Now, for the purposes of this talk, I want you to remember that there are two flavors of NB-LRR proteins. There are those that encode an N-terminus domain called a TIR, and those that do not. So I'll refer to these as TIR and non-TIR. So both of these are NBLRR proteins, but there are these two different lineages that we know are monophyletic, and these are really an ancient distinction. And it'll become important later on in the talk. Now, at a phenotypic level, these R proteins are playing the role of cellular sentinel and cellular switch. So the proteins are constitutively expressed at very low levels, and the LRR in particular, through protein-protein interactions, recognizes the presence of a pathogen effector, and by extension, the presence of the pathogen. So the LRR plays the role of cellular sentinel, and once the LRR detects the presence of a pathogen, a conformational change in the protein that's mediated through the NB domain results in activation of a downstream cascade that results in the phenotype that we see as disease resistance. And so these NB-LRR proteins, and particularly the NB domain, serves that role of cellular switch to activate defense responses. And this can include things as divergent as the hypersensitive response and expression of PR proteins. Now, while our understanding of how plants recognize and respond to pathogens has really blossomed in the last 20 years, so too has the availability of whole genome sequence for plants. And this is a timeline showing that the very first plant to have its genome fully sequenced was Arabidopsis back in 2000, followed very quickly by the genome of rice in 2001. So for a very, very long time, Arabidopsis and Rhiza were the two genome sequences we had being the models to represent all dicots and monocots respectively. But the rate of discovery as new DNA sequencing technologies have emerged and as new informatics approaches have emerged, has really sped up. And by 2017, 250 plant species have been fully sequenced. So the rate of discovery really is very important. For this particular talk, we'll focus on the Rosaceae. And there are three species that were really fundamental in some of our early work. So the genome sequences of apple, Fragaria vesca, which is a diploid relative of the strawberry, and Prunus persica, or peach, were the first three genomes that were fully sequenced in the Rosaceae. These were done at a very high level. And today we have a total of 15 different Rosaceae species that have been fully sequenced. So the Rosaceae is a really uncommonly, as a family, it has a really uncommonly rich genomic resource for analysis. Now, it might surprise you that given our robust understanding of how plants recognize and respond to pathogens and the robust genomics resources that we have available to apply to plants, might surprise you that comparative genomics approaches are rarely used to study disease resistance. So comparative genomics are using the information we've gained from one particular species to understand how another plant species functions. It's really revolutionized so much of plant biology, but it's almost never applied to NB-LRR genes. And there are at least three reasons why this is the case. First of all, there are many, many R genes that are encoded in every plant genome. So this table is summarizing our analyses of 12 different rosaceous genomes. So on the left, there's Rubus occidentalis, the blackberry genome, and on the far right is Malus domestica, or the apple genome. So you'll see that there's wide variation in the total number of R genes that are encoded in these plant genomes. And the numbers range from only 43 in blackberry, which is just ridiculously small. The vast majority of plants have several dozen to several hundred R genes. So 43 is a really small number, to more than 660 in apple. So there are both a lot of R genes and a wide variation in the number of R genes that are encoded in plant genomes. So this is a complication for applying comparative genomics approaches. Secondly, R genes evolve very, very quickly. And this slide comes from my colleague Nevin Young. Nevin and his group for many years has worked on a model legume known as Medicago truncatula. And one of the projects that his group has led is the sequencing of a large number of wild genotypes of Medicago truncatula. And here he's summarizing the composition of multi-gene families across those genomes. So the x-axis is a measure of synonymous changes, so changes in DNA that do not correspond to changes in protein sequences. while the y-axis is a measure of non-synonymous changes, so changes in DNA that do equal changes in the encoded protein sequence. The thing that we need to understand to interpret this graph is that anything that's down in this quadrant is relatively slow evolving, while anything up here is very rapidly evolving. And amongst the most rapid evolving components of plant genomes are the TIR, the NDR, and the LR. So these three components that we see recurring over and over again in our proteins. This particular gene is late nodulin. It's not an R gene, it's not an NDLR gene, but it is involved in mediating legume-rhizobium interactions. So in general, we see that the genes that are involved in plant microbe interactions develop very rapidly compared to other components of the plant genome. So this rapid rate of evolution, while it makes good sense from a biological perspective, really complicates our ability to apply comparative genomics. A third challenge in using comparative genomics to study our genes relates to the chromosomal organization of these genes. So we've already talked a bit about the composition of individual genes, but it's very rare to find an R gene as a single locus on a chromosome. More often these genes occur in clusters of sequence-related but functionally divergent gene copies. Now this clustering is really very important for creating new allelic variation at these loci that helps plant genomes evolve more rapidly at these loci to keep pace with changing pathogen populations. And through intra-locus recombination or intercluster recombination, we see that the clusters themselves can really expand or contract very rapidly, and this expansion contraction equals the formation of new allelic variants, some of which would be effective against pathogen populations. So biologically, this is very important, but this rapid evolution also obscures the one-to-one orthological relationships, or the common ancestor relationships of these genes, which really is a violation of typical comparative genomics approaches. Now, to give you a visual of what this clustering looks like in a real plant genome, this is the genome sequence of Prunus persica, or peach. This comprises 8 chromosomes, and we've arrayed them here end to end. And each of the bars that you see represents a different R gene that we've discovered in that genome sequence. So just looking briefly across the genome, you see that R genes are not spread equally throughout the genome. They, in fact, there's a very strong tendency for these genes to cluster in certain common regions. So all of these are challenges to comparative genomics. We talked a bit about the fact that plant genomes comprise many R genes. These genes evolve very quickly, and that the R gene orthological relationships are quickly obscured. But I'm here to say that I believe there is both an opportunity to apply comparative genomics to the study of MDLR genes, and also I think a really important agricultural application for doing so. And the work that we're doing is in the Rosaceae. I think this is a plant family that I would guess most people are familiar with. Horticulturally very important. A lot of our fruit crops come from this particular plant family. And of course, rose is the genus, or the Rosa is the genus that gives name to the family. So that's in the Rosaceae as well. I've already talked a little bit about the very robust whole genome sequence data that are available for this particular family. Another attribute that is valuable in our research is the robust phylogeny that's been developed for this family. And this family, this phylogeny also has a key chronological component that allows us to ask and answer some very sophisticated evolutionary questions. So we know that all of the species that we today refer to as Rosaceae emerged from an ancestor that lived 100 million years ago. We know that rose, blackberry, and strawberry emerged from a common ancestor 59 million years ago. The pomes and the stone fruits emerged 52 million years ago. Apple and pear diverged 32. A common monophyletic origin that is more recent than 32 million years ago, but we don't know exactly when it is. So this phylogeny, and particularly the chronological component of this, really is a key piece for some of the evolutionary analyses that I'm about to present. So the work I'm sharing is largely the work of Dr. Leon Van Eck. He was a postdoc in my group up until last January. He's now the one and only assistant professor of plant biology at Augsburg University in the Twin Cities, so we're very proud of him. And a lot of the conceptual biological focus of the research comes from Leon and my collaboration with Leon. More recently, Dr. Greg King, who's a computer scientist, has been working with us to take the biological concept and to create custom Python scripts that allow us to input whole genome sequences and output what I think are biologically meaningful observations. So key to our approach is recognizing that the R gene clustering that is so critical to the biological function of R genes really represents a degree of phylogenetic redundancy. And what I mean by that is that all of the gene copies in this cluster trace back to a common evolutionary origin. And we know that this divergence of this cluster, of the individual copies within this cluster, is a relatively recent event. So these are, this has allowed us to create what we think of as composite R gene sequences, or apply a diversity binning approach that allows us to reduce the overall phylogenetic allelic diversity at these loci into a composite gene sequence that represents the entirety of that group. Now, I like to think of this as akin to the species concept. So we recognize as biologists that the species concept, number one, is an artificial construct. We also recognize that the species identity is really biologically meaningful. And we understand that the various genotypes that comprise a species are not identical, but that the species idea represents a limited range of genetic diversity, and that we can talk about species as a whole, as a group. In the same way, we think of these composite groups as representing the genetic diversity of a small but related group of DNA sequences. Now, to facilitate dialogue around this, we've also created a nomenclature. We refer to these groups as Roser 80 lineages. Roser 80 stands for Rosaceae R genes at 80% identity. That 80% identity comes from the fact that we're applying at a protein sequence level an 80% cutoff in the NV region to define those diversity clusters. So if two sequences are at least 80% identical in the NV region, they're collapsed into a Roser 80 group. And we've done a lot of empirical testing. I'd be happy to chat with anyone who wants to know more about why we chose 80%. But we think this is a biologically meaningful cutoff. So in this study, the graphics I'm about to show you, we've looked at 12 different species from the Rosaceae. Our custom script has applied a uniform definition for what we're calling an R gene. And across those 12 species genomes, we've identified nearly 2,500 R genes. And we've been able to collapse that diversity to 651 Roser 80 groups. So 651 is still a pretty large number, but it's small enough that we can begin to visualize patterns of evolution over these 12 different genomes. And really what we're doing largely is an exercise in data visualization. So one of the outputs of our script is a phylogenetic tree. So this is a maximum likelihood tree that comes, that represents these 651 Roser 80 groups. So each of those branch points is one of those Roser 80 groups. This tree then allows us to annotate with other forms of data. So first I'm going to layer on representation of R genes across the 12 different species. So each of these concentric circles represents not an individual species, but actually a tribe. So I could include 12 concentric circles going out, one for each species. It gets really, really complicated very quickly. So instead on this graphic, I'm showing things at a higher taxonomic level known as the tribe. So the inner circle, represented here by this very deep purple, is Rubiae. In this study, we have a single genome sequence from Rubus occidentalis, the black raspberry that is in the Rubiae. Next, we have in red the Potentillae. Potentillae includes cultivated strawberry and its wild relatives. So we've got the genome sequences of 6 different gregarious species in this study. The sort of peachy color, the orange here, is the Amygdalae, which includes the stone fruits. And we've got 2 different prunus species, that of peach and Japanese plum, represented here. And then the outer circle is the Pyrrhae, that's shown in yellow. That includes apple, the European, and the Asian pear in this particular study. So anywhere you see a colored box, it indicates that the corresponding Roser 80 group has a representative from that particular tribe or species in that particular genome. So immediately we're starting to see the distribution of our genes across really 100 million years of evolution in the Rosaceae. We can layer other types of information on there, such as a topological definition of clades. So that's shown up here. It also is reflected in the color of the tree itself. In this study, we identified 9 different clades, and we've labeled these A through I. And it's very interesting to note that those clades themselves can be grouped into TIR and non-TIR types. And a moment ago, I told you that there are these 2 different flavors, that we know these are of monophyletic origin. And so the fact that we're actually recovering both TIR and non-TIR clades really serves as almost an internal control for this study, telling us that this idea of diversity binning and application of this phylogenetic strategy really is giving biologically meaningful information. So this represents 12 different genome sequences in the Rosaceae. I've already mentioned there are a total of 15, so we're working now on Rose. The Rose genome came out last year, and I'll show an analysis of this in just a moment. Prunus avium, sweet cherry, was published last year as well, and very recently Potentilla, which is related to the Fragaria species, has been published. So the pace of discovery of creating whole genome sequence in the Rosaceae continues to expand, and we're working to update that image that you saw just a moment ago with some of these new sequences. Now this figure, while relatively simple, took many years and lots of thinking and lots of scripting to get. But now that we see this, now that we have this sort of global view of our gene distribution across the Rosaceae, we really can use it for both evolutionary observations and for applications. So I want to give you examples of both evolutionary observations and some of the agricultural applications of this tool. So first of all, our gene lineages are of ancient origin, and by that I mean that this magical ancestor that lived 100 million years ago that gave us all the wonderful things that we think of as Rosaceae today was challenged by pathogens and already had representatives of most of those 9 clades of our genes. And of course, as that species evolved and speciation events led to new morphologies, new physiologies, new pathogen pressures, the R gene component of the genome continues to adapt until we, out here today, have an extant collection of R genes that most of which date back at least 100 million years. So we're really looking at a series of evolutionary events that correlate with the speciation throughout the family. Second, we can see from our tree, and this is sort of a corollary of the last point, everything is everywhere except when it's not. And so by that I mean that most of those R gene lineages have ancient origin, but it's a little more complicated than that. And I think it's the exceptions that are particularly interesting. And I'm going to show this a couple different ways. It's a little difficult to show, so there will be a couple different ways of looking at this. This figure is best read from right to left. So the right column here represents the TIR and non-TIR R genes. We have next mapped those to the 9 different clades that we've defined, A through I. And finally, we've mapped the presence of these 9 different clades onto the 12 different genomes that went into this analysis. Now, if you stare at this really intently for a very long time, certain patterns are going to emerge. First of all, I want to point out clade F. It's this tiny, tiny little clade here. It's actually represented in our study by a single Rosardae group, and it's found only in the Pyrrhae, so it's only found in apple and pear. So the most parsimonious interpretation of this is that that R gene clade emerged in the pome fruits relatively recently. Now, in contrast, clade A is really one of the Rosaceae species, but we don't find it in Prunus persica, and we don't find it in Prunus mume. Now, when we first did this analysis with only Prunus persica, I thought, oh, there's something wrong with that sequence. Maybe if a different peach variety was sequenced, or maybe if the genome build was updated, we would suddenly find representatives of that. And then came along the Prunus mume sequence, and in fact, again, we found no observation of clade A in that species either. So the most parsimonious interpretation of this is that clade A is of ancient origin. It's 100 million years old, at least, but has been lost in the stone fruits. Now we can make these same points using our original tree. Clade F is this thin blue line represented here, again found only in apple and pear. We think it's probably of recent origin, meaning it's no more than 32 million years old. And in contrast, Clade A probably was lost in the amygdalae in the prunus species no more than 52 million years ago. Now, I mentioned a moment ago that a third prunus species genome is now available. We've actually done the analyses. I don't have the pretty picture to share with you today, but I can summarize our finding. In fact, Clade A also is absent in the genome of prunus avium. So it's beginning to really look like this particular clade of our genes was completely lost in the stone fruits. I really need to highlight that this figure and some of the ideas I just shared with you are hypotheses. I think one of the powers of what we're doing this comparative genomics approach is creation of hypotheses. We're beginning to see patterns across millions of years of evolution that would not be obvious if we were only working in one particular genome. Obviously, we want to follow up on some of these observations and test these hypotheses. Now, one other example of this exception to the everything is everywhere rule comes from Fragaria species, and we have 6 different Fragaria species or strawberry species that are included in this study. These 6 species are represented as columns in this table, and the 9 argine clades are represented as the rows. There are a couple types of data that we're reflecting here. I think the main thing you need to understand is that if you see a circle in any particular cell, that means there are argines from that given argine clade found in that particular species genome. And really the larger the circle and the darker red it is, just means there are more argine sequences found in that genome. So a couple patterns jump out. First of all, clade F is completely missing in Fragaria. Clade F is that apple pear only clade, so that makes good sense. But if we look at this particular species, this is Fragaria vesca. Fragaria vesca is by many considered to be a model for the Rosaceae for many applications. So I'm really disturbed to see that we have no representation of argine clade A, argine clade D, or argine clade E in the genome of the sequence. Now there could be technical reasons for it. There could be biological. Maybe if we sequence a different Fragaria vesca genotype, we'll find those argine clades. But I think this really is a cautionary tale for the overuse of models. And what I want to advocate for is lots and lots of genome sequences of lots and lots of different genotypes from lots and lots of different species. So that's really where I think we're headed in some of this work. Now, a third evolutionary observation that emerges from our data analysis is that patterns of argine homology reflect species phylogeny. I'll explain what this means. Now, as a reminder, key to our approach has been collapsing this phylogenetic redundancy into these composite sequences that we call the Roser 80 group. And importantly, we did not constrain species affiliations. So a Roser 80 group can be comprised of multiple sequences from one species, but it can also be comprised of sequences from multiple different species. And what we find is that the more closely related to species or to tribes is, the more likely they are to share genes. And that actually makes good sense, but it really is the basis of phylogeny. Now, this Venn diagram represents the composition of our Roser 80 groups across the various different tribes. You see that the vast majority of Roser 80 groups comprise sequences from only one tribe. And that makes sense. We know these genes evolve rapidly in response to pathogen pressures. And so that adaptation, that pattern of adaptation is reflected in those numbers. But where Roser 80 groups are comprised from sequences from multiple different tribes, we see that more closely related tribes share more genes than do the more distantly related tribes. So this makes good intuitive sense, but really, in my mind, is the justification for using a comparative genomics approach to study our genes in the first place. Now, there's another really interesting observation that comes out of this comparative analysis, and that is that we've identified a handful of very slow evolving R genes shown right here. So these R genes are found in all 12 of those genome sequences. Some of these species are separated by 100 million years of evolution. And the fact that they're shared across these species means that the NB domain is not diverged by more than 20% in 100 million years. Right? So this does not match our understanding of how our genes evolve and what they're actually doing. And I think that's a really interesting observation. Note that this would not be possible. We would not know this if we were only studying the genome sequence of a single species. So this comparative approach really is giving observations that I think are meaningful in biological context. And if we have time at the end, I'm going to come back to this because I would love to pick your brains on exactly how to test what these genes are doing. Now, our final evolutionary observation is that species differ in patterns of our gene diversification. I think this is the most important observation and is really our entree into hypothesis testing and agricultural application. So when we look across the tree, we see that while most of these clades are well represented across all of the species, there is a tendency within a given clade for certain species to have an over-diversification of alleles. So, for example, in this subclade found in clade E, we see apple and pear have lots and lots of copies of our genes from that subclade, but there are none in the other rosaceous species. So the fact that apple and pear are making good use of this subclade means something in a biological context. There's something that this group of genes is doing for apple or pear that is irrelevant to strawberry or rubus or other species. Now, there's another layer of data that I want to share with you, and this is a heat map that represents the composition of the individual Roser 80 group. So the Roser 80 group is a cluster of genes, so a cluster in this context is at least 2 sequences that are grouped together. But in this study, we found the composition of individual Roser 80 groups range from 2 sequences to more than 60 sequences. And the heat map reflects that, so a darker color indicates 2, and white sequences, you see one right here, for example, represents 60 different copies of it. This heat map will be valuable in just a moment. So from an evolutionary context, what we've learned from this data visualization approach is that our gene lineages are of ancient origin. Everything is everywhere except when it's not. Patterns of our gene homology reflect species phylogeny, and that species differ in patterns of our gene diversification. Now, where the rubber really meets the road, though, is in hypothesis testing and the use of this Roser 80 system to visualize allelic patterns and to use it to identify useful traits for crop improvement. One of the key questions I've asked myself over the years is, has agriculture escalated the molecular arms race between plants and pathogens? And we know from a robust literature that large-scale monoculture and modern agricultural practices have really influenced the effector component of pathogen genomes. And we see in particular effectors adapting to large-scale monoculture through effector suites that really represent or are effective against the predominant plant genotypes. I think the exact same thing is happening in the plant. I think it's manifested in a very different way. It takes into account how our genes actually evolve. So how do we get here? Well, to test this idea, we've turned to apple scab. Apple scab is a disease of apple, scabby disease of apple, caused by the fungus Venturiae inequalis. It has long been a target for researchers and breeders. At a phenotypic level, there are 20 different disease-resistant genes effective against Venturiae inequalis that have been defined. Only 2 of these have been cloned. They both are NBLRR genes. And in this study, we've called the literature to identify anything. So if I'm right that large-scale monoculture has led to pathogen populations that are well adapted to the predominant plant genotype, and if I'm right that pathogen pressures imposed on agriculture have influenced plant genomes in a way that corresponding R genes undergo an allelic diversification process, this should be true. And to cut to the chase, when we looked at our data set of 15 candidate R genes, clade E becomes particularly prominent, with 3 of those R genes mapping to subclade E, that subclade that is uniquely diversified in apple and pear. And in fact, RBI1, RBI4, and RBI15 reside in Roser 80 groups that have somewhere in the neighborhood of 30 or 40 gene copies. So this pattern that we're seeing is consistent with an idea of iterative evolution, where Apple has hit on an R gene that is effective against Venturi and equalis, and then through the mechanisms of cluster expansion and contraction of R genes, the apple has created new allelic variants at those loci, and some of those loci, those new allelic variants are effective against Venturi and equalis, and some are not. But the expansion of those clusters is evolutionary footprint indicating that that subclade is important, and in this particular case, we can demonstrate that the apple scab resistance genes preferentially occur in those highly diversified subclades. And in fact, I'm only showing 3 of these, but we saw very strong statistical support for that idea throughout the apple genome. So in this case, we're beginning to understand in an evolutionary context what an apple scab resistance gene looks like, or more correctly, to understand what an agriculturally important resistance gene looks like. We don't necessarily know the function, but we do know that this subclade, because it is highly diversified in apple, should have some meaningful applications for apple. This opens a new opportunity to apply allelic mining of gene bank collections to inform phenotyping and trait discovery. Now, to accomplish this, or to test this idea, we're partnering with Professor Stan Hokanson at the University of Minnesota and Professor David Zlizak at the University of Wisconsin River Falls. I've worked with these 2 guys for at least 10 years on various projects in rose. So Stan is a rose breeder. He's a co-PI on the USDA CAP project known as Rose Breed, the focus of which is to develop markers linked to disease resistance genes for improvement of rosaceous crops. So we've generated a large segregating population of disease-resistant or black spot resistant roses. We have completed phenotyping. We've done a lot of SNP marker analyses already, and we're now expanding that data set to include some of the analyses from the Rose 80 group. This is really a placeholder just for me to indicate that we have already initiated our analyses of the rose genome, running that through our pipeline. In this case, we're applying it at a subfamily level, comparing rose, Fragaria vesca, and Rubus ascendentalis. So the Rose 80 pipeline is applicable at the family level, but also at some of the subfamily levels. So our objective in this study is to leverage the Rose 80 system to develop perfect markers for marker-assisted breeding in rose. And for that, we're using a DNA capture approach that was really pioneered by Jonathan Jones's group at the Sainsbury lab, ironically enough, in potato late blight. So the idea is to use what are called baits, so DNA baits that represent the R gene components of the rosaceae in this particular case. These baits are then applied to disease-resistant and disease-susceptible DNA bulbs, and the baits are used to pull down corresponding genome sequences, corresponding but not necessarily identical genome sequences from the DNA in solution. This then creates a pool of R gene-enriched DNA that can be sequenced at large scale. And our goal here is to sequence these libraries and to achieve allele-level resolution that allow us to identify candidate polymorphisms and, of course, to validate on the larger mapping population. And so if we're successful in this effort, we will have markers that are derived from the allele that makes rose black spot resistant, so that becomes a perfect resource for marker-assisted breeding. More importantly, this really serves as a proof of concept effort for larger scale mining in rose, but we can also apply the same idea to the Venturia inequalis or apple scab pathosystem or potato late blight, or really I think any plant family or plant pathosystem. How am I in time?
You're good, about 5.
All right, I'm going to zip through this because this is really cool and I really want your input on this. I pointed out that we identified in this analysis a small group of R genes that are very, very slow evolving. And I have some thoughts on what these things might be. I'd love to hear your thoughts and any approaches you might have to actually testing this. So it's possible that these R genes are still disease-resistant genes, but that they play a role in non-host functions. So maybe they're so good at fending off a would-be pathogen of the rosaceae that we don't even think of that microbe as a pathogen on the rosaceae anymore. And that relationship has been so stable that these R genes have really not diverged much in 100 million years. It's possible that these R genes are not involved in disease resistance per se, but are involved in maintaining positive plant microbe interactions, which should be stable over evolutionary time. It's possible that they no longer retain a disease resistance function at all. Perhaps these genes have played some role in environmental stress adaptation. Now, a few weeks ago, I would have stopped the talk right here, and that would have been as far as my laundry list of ideas would be. But more recently, we started applying the Rosarini concept, this comparative genomics approach, to the disease resistance genes in the Solanaceae as well. Now, in the Solanaceae, we've got a total of 7 genome sequences that we're working for, ranging from tobacco to some of the peppers to, I can't even read that, wild tomato, cultivated tomato, wild potato, and potato. We also have a collection of 15 cloned NV-LRR genes that we know to be effective against late blight. So that's all of those data are shown in this particular figure, with the black bars that you see here representing Rosarini groups that comprise the late blight resistance genes, followed by the cultivated potato, wild potato, cultivated tomato, wild tomato, and then the more distantly related capsicum and tobacco. What I want to point out is that two of the identified late blight resistance genes have closely related homologs in every one of those genomes. So that means that, similar to those 4 slow evolving genes, that these particular genes are very, very slow evolving. I think the relationship here between these species dates back only to about 30 or 40 million years ago. But the late blight resistance genes fall into these clusters, and some of the other late blight resistance genes are represented at least in the majority of these genomes. So we know that Phytophthora infestans causes late blight in tomato and potato. We also know Phytophthora infestans is not a pathogen of capsicum or Nicotiana. So what this pattern indicates most likely is a long coevolutionary relationship between the Solanaceae and Phytophthora that really predated speciation events in both of these taxa. So this allows us to add maybe another potential explanation for those conserved R genes that we find in the Rosaceae, and that is a pattern of coevolution. So it's fun for me to look at this, to visualize these conserved R genes, what they're doing biologically. I can only sort of imagine, but if you have ways of testing any of these hypotheses, I can overexpress or knock out a gene. I don't know what the phenotype is. I don't know what pathogens to challenge a given plant. So if you've got some creative approach, I'd love to hear your thoughts on this. So just to wrap up this talk, I presented a comparative genomics approach to analyzing evolutionary relationships of our genes. We've identified a number of evolutionary observations that these lineages are ancient, that everything is everywhere almost. Our gene distribution reflects species phylogeny, and that species differ in our gene diversification. And this observation in particular lends itself to agricultural application. And in particular, we're generating hypotheses that are beginning to link sequence diversification patterns to specific disease resistance function. And of course, the end goal of this is the potential to mine gene bank collections for crop improvement. Thoughts or questions? I'd love to hear them.
Questions for Dr. Bradeen?
Or thoughts on how to test those conserved R genes? Yeah.
Occurs to me that as you get more phenotypes for this last hypothesis, a lot of it will fall out. Because at MLA now we've seen related orthologs for wheat stem rust, wheat stripe rust, barley stripe rust, barley spot blotch, rice blast, and a few others, all occurring at that one locus.
So really allelic variants.
Yeah, so it was originally coded as MLA, but now it's been shown, ortholog's been shown to code for UG99 resistance and a number of other things with rust and the rice blast and Victoria blight, that's the other.
Oh, okay.
So these are a number of important diseases over the years with cereals, and they're all coming back to roost at one locus. So to me that actually looked very similar to this, except you just don't have a phenotype yet.
Yeah, and you might very well be right about this.
So you have all the genetics, I mean the genotypes, but you don't have the phenotypes. So it took a long time to evolve this little piece of knowledge that I just told you. I mean, across a lot of groups. So I think as you get more phenotypes, the different hypotheses will bear themselves
Shake out
out.
a little bit more.
So that comes back to your first thing, genotyping is more, you're focusing more on genotyping and phenotyping. I think you need to combine them both.
Yeah. Oh, I totally agree with that, by the way.
Yeah.
And I don't want to give, I think I actually neglected to explain how we see this being applied, that doing large-scale genotyping of our genes in gene bank collections is really the first step. The next step would be to identify novel allelic variants at these loci and use that information to identify genotypes and populations or species that would be subjected to more intense and hopefully more informed phenotyping approaches for drug discovery. So this doesn't negate at all value of phenotyping. It's meant to inform that process.
So my guess would be that these different forms actually encode some different disease resistance.
And it could be. You might very well be right about that. And I think I sort of glossed over some of the early pioneering work of Molly John's group, when she was still at Cornell. They actually looked at phenotypic resistance and what had been reported in the literature as phenotypic resistance across capsicum, tomato, and potato genomes, and found that that phenotypic resistance maps to corresponding genome locations across these. And that they noted a lot of divergence in terms of what a particular locus is doing. So it's still involved in disease resistance, but in tomato, for example, it might be a virus resistance, but in potato it's nematode, and in pepper it's an oomycete. The exception, though, to that rule was that Phytophthora resistance tended to map to the same location, even in capsicum. So capsicum is a host to Phytophthora capsaicea, which is, of course, a Phytophthora species. It's not a host to Phytophthora infestum. So that was the first indication that there's probably this long coevolutionary relationship between this. I think we're seeing that same pattern here at a finer scale resolution. And so what you're describing in grasses, I think, really matches what we're seeing here too. So my guess is that if we want to understand what those genes in Solanaceae, for example, are doing, what a gene in tobacco or a capsicum is doing that we know in potato with a late resistance gene, I think we should be looking for Phytophthora resistance as the first phenotype to survey for.
Would you expect the variability in the rate of mutation differences in these R genes to differ between long-lived perennial wood species and the shorter-lived species?
What a beautiful question.
And if it isn't that different, why would you have all those R genes in these long-lived woody crops?
So,
because they're not doing, it's not a red queen hypothesis.
no, it's an excellent question. And there is this idea in the literature that tree species should have more R genes than non-tree species because they're long-lived and would have fewer generational opportunities for recombination. I have a hard time wrapping my head around this. I actually traced that comment to the sequencing of the poplar genome, so it's stated in that Nature paper, I think it is, without any reference. It's just sort of a comment that was made there. It's been repeated many, many times. I mean, okay, I don't really buy that. I don't see the evolutionary dynamic that would cause a tree to have more R genes than an herbaceous plant. But I'll be damned if we aren't looking at the Rosaceae. We see that apple, pear, European pear, the other way around, but Asian pear, European pear, and the stone fruits have more R genes in this analysis than do Fragaria and rubus. So, I mean, that actually fits that pattern. I'm still not convinced. What I think is happening is that cultivation and in the intensity of cultivation of a species is driving the number of R genes. So a cultivated plant in general, and especially one that's cultivated at large scale, in my world thinking should have more R genes than a wild relative. We are nowhere near having the data set to actually test that hypothesis or what you described as tree species or long-lived species having more R genes. So I really don't know.
Maybe I just missed the point of what you said, but cultivation hasn't been going on for very long, and you're looking at 100 million years of diversification, so, I mean, I don't see that cultivation would necessarily be a stronger force or...
And I think I would have agreed with that a few years ago. So my thinking about R genes has really changed a lot, and I don't know, I mean, this is more thought experiment than data at this point. I think that clustering mechanism of R genes actually allows very, very rapid adaptation, expansion, contraction of R gene clusters in plant genomes. So I think almost in real time, if we have the technologies, we could measure that. So if I could sequence individual pollen grains, I think I would see variation in some of these clusters. If not that scale, then maybe over a 10-generation scale. I used to think, it would take millions of years. I think it actually happens a lot faster than that. And I could be wrong, but at least the data that we've been able to test in terms of apple scab resistance really fits very nicely with this idea that even short, relatively short, comparatively short periods of cultivation have been enough to really influence the representation of alleles across the genome. Yeah.
Just one thing I wanted to add. That recently it's been reported that in an unselected field of barley and clone, they actually can recover ADR effectors at a relatively high rate because they're maintained in an unselected field in an unselected population of mildews. So that kind of goes against, not against, but kind of puts a kind of a chink in the hypothesis that cultivation of the widespread selection for these types of, I don't know. Anyway, I just want to bring that up because it was interesting because I just thought that you don't even have to look at worldwide collections. You just go into an unselected field and find what you want because it's all maintained there.
Because it maintains the population. Yeah, and we see the same thing actually in Phytophthora infestans. So an R gene that's defeated once is pretty much gone in terms of effectiveness.
So are you saying there's more R genes because in cultivation because people are selecting for resistance?
Not necessarily. So it could be in part intentional selection on the part of breeders to breed apple scab resistant trees. So I would expect that to lead to an expansion of particular R gene clades that are effective against Venturi and Equalis. I think unintentional selection, so I don't want to eat the scabby apple, I'm not going to save that one. That actually could have that same impact. I also think that without human intervention, a pathogen that's really good on a particular monoculture and wipes out a field is probably going to have a similar impact.
You showed that wild potato relative that's resistant to everything.
Right.
And then I would have thought starting wild, I thought the standard story was then people bred for nice, big, tasty potatoes. Like sorts of things.
They really are.
They weren't bred for resistance.
And I think just because there's a high level of allelic diversity in a cultivated genome doesn't necessarily mean that it's effective against the predominant pathogen population, right? Because the pathogens are always evolving, and we're, by planting large-scale monoculture, we're more or less favoring that. So I don't necessarily think that translates directly into phenotype. But what I do think, what I suspect, and I'm being transparent, but this is all hypothesis, right? And this is a thought experiment more than anything. I do think that while the pan-R genome, so the entirety of the R genes that we find in a wild species, will be far greater than what we see in the cultivated species. But I don't think that translates into an individual genome. So on average, I would expect the genome of a single wild plant to have fewer R genes than a genome of a single cultivated plant. Now, there's tremendous variation in the number of R genes, right? So this is not something that we can go out and sequence one genotype of a wild species and one of a cultivated and say, aha, here, we've proven anything, right? This is something that we're going to have to, over many NSF grants, I hope, be able to actually generate the data that we need to test this. Whether that's true or not, I think there are a lot of interesting observations that are going to allow us to link some of these evolutionary patterns to biological function. And that's really where I want to go with this.
questions? Well, thank you.